Hello everybody and welcome to the second tutorial in our Docking with Fitted series. My name is Mihai and today I will be discussing how we do flexible docking with Fitted. For today's tutorial, you will need the Drug Discovery Platform Forecaster, which you can download for free if you are in academia. Otherwise, if you are an industrial company, you will require an industrial license. A visualization software such as JMol or Biovia Discovery Studio, a text editing software like Notepad++, and a web browser, for example Mozilla Firefox. The recommended readings for today's tutorial are papers 3 and 7 in our series of docking ligands into flexible and solvated macromolecules, and I've linked here the title of the paper and where you can find it. They're both published in the Journal of Chemical Information and Modeling. The first one is published in 2009 and the second one in 2014. As an overview, I've linked a good review published in the Briefings of Bioinformatics approximately 4 years ago. The key terms that I'll be using in today's tutorial are flexible docking, conformational ensemble, superposition of protein structures, and again the protein data bank. With this being said, let's start our tutorial. In today's tutorial, we will use the same protein that we used for our rigid docking tutorial, and that is the structure of the Herpes simplex virus type 1. In our rigid docking tutorial, we used only one protein structure, and that was 1E2K. However, there are multiple other protein structures that can be used for a virtual screening campaign. These protein structures are 1E2P, 1KI3, and 2KI5. The reason we would choose to do flexible docking over rigid docking is whenever we have multiple protein structures that all contain some different information, especially flexible amino acid side chains, we'd prefer to use all this information and not discard it. So what we do with Fitted is we use the information from all these protein structures to dock molecules to this target. So what we'll do is we'll first open our platform. We will go to our current working directory, which is right here, flexible docking, and we'll click on our preset workflow just like we did in rigid docking. However, in this particular tutorial, we will be making use of matchup. Matchup is a module that superposes different protein structures, and in the case they contain different or mutated residues, it mutates them back to their original residue. So what we're gonna first do is we're gonna download the protein structures that I've just described here. So we're gonna download 1E2K and download PDB. Then we're gonna download 1E2P. And then we're gonna download the other two, 1KI3 and 2KI5. Alright, then we'll open our folder and you'll see we have all the protein structures here. So for now, I'm just going to include a matchup to show you what the module does. So when we open it, you'll see a directory that shows you the number of proteins that you would like to superpose. So we would like to superpose 4 different proteins. These are 1E2K, 1E2P, 1KI3, and 2KI5. Here you can put either all or just a specific side chain. We're just gonna keep all for now. And then what we're gonna have to do is we're gonna have to load the PDB structure, and we're gonna have to choose the ligand that is bound to these to all these protein structures. So for the first one, for 1E2K, we're going to use the exact same ligand, TMC A500, that we use for rigid docking. We click on select ligand residues, and then we write the keywords, and you'll see the keyword has appeared here. Then if we try to open a second window while the first one is still open, we get an error message, so we have to first close this one. Then we load the second one. We're gonna choose this one, CCVA500. Select the ligand. It's a bit tedious having to do it multiple times, but it gives you a very good practice on how this workflow goes. This was a PE2. We do the same. And then the last one is AC2. So once this is done, we can put the output file name, which is step 1 superposing proteins. And then what we're going to do is, there are two different modes here. One of them is superpose, which just superposes the proteins and does nothing else. And the other one is make similar, which makes sure that all the mutated amino acids have been reverted to the original amino acid. So we're gonna choose make similar in this case. So then I guess I should change the output file name for this, make similar proteins, and then we save. Once this is done, we exit, and now you see the little check mark, which means we've set up matchup properly. And then we're just gonna run the workflow with this specific module, because I want to show you exactly how the module looks like and how it works. Alright, so here you see the different superposition of proteins. I think I just closed my terminal. I apologize for that, but I can just open the output file. So here you will see for each of the proteins, they're in the same order that you chose them in the GUI. So this would be 1E2K, then you would have 1E2P, 1Ki3, and then 2Ki5. And at each position it tells you what amino acid you have. So methionine, proline, threonine. And then so you can go down, and then if there are any gaps, it will show it as a gap. So for example, which one is this? 1E2P in this case has a gap, an amino acid gap between 69 and 75, so amino acid 70 is missing. You can go down just like this, and then it will tell you what the RMSD is between the different protein structures. The lower the RMSD, the closer in shape the protein structures are, and then it gives you an idea of the residues it mutates and deletes. So you can keep track of what matchup has done. And then at the end, you get the common ending message. So now in the folder where we're working, in our working directory, you're going to see 4 different protein files called 1E2K underscore aligned and mutated. If you would have done only superposition, the name of this file would have just been underscore aligned. So by doing the make similar, by choosing the make similar mode, you get this aligned and mutated mode. So now we can visualize these proteins in a visualizer. You can actually, I'm going to show you a trick, because I think it's I didn't show it last time, and it's something that's very very useful. You can actually click on the magnifying glass right here, and it will open the results menu. Basically, you can check the job completion, and by clicking on this, if the file names appear here, it means that your job is done. Then what you can do is you can load the structures, and it will load all the structures into JMol, and you can see the 4 different protein structures superposed and where the ligands are. So you can do this in the native JMol menu if you don't have your own preferred visualizer. I do it in Discovery Studio because I've just gotten used to Discovery Studio, and it's very easy to use. So I'm just going to show you. For now we have 1E2K, so let's open all the other ones. 1Ki3 and then 2Ki5. So then we'll just copy and paste the structure from each one. So you can see they're very nicely superposed. In this way you can also check where the flexible loops are. So for example, here there's a little loop that is different between the different structures. You can, with pretty high confidence, check to see where these flexible areas are and if they're close to your binding site. For us, the binding site is here, and the differences aren't very very pronounced. So now that we have these structures, what we can do is we can close the results there. We can go do our normal workflow. First, we'll select prepare to prepare the protein structures for docking. So because we have 4 different structures, we'll be working with 4 proteins. This gets automatically updated from the matchup box above once you select it here. So you don't really need to change anything. These are proteins, they're non-metalloproteins, so we're just gonna keep the proteins here. And in this case, in this specific tutorial that we're doing, we're actually going to use our own ligand. We're not going to use the ligands to do a sort of pseudo self-docking to 4 different structures. We're going to use our own ligand and see how well fitted docks it to the 4 different structures. And for this, we're going to use the sketcher, convert, and smart. So we're going to use the entire workflow for this tutorial. So the sketcher is a tool that we developed to make it easier for medicinal chemists, because it's very similar to ChemDraw. So this is the ligand that we're going to use today to do our docking. I'm just gonna draw it here. You can use the same sort of shortcut, like the keyboard shortcuts that you would use with ChemDraw. Obviously, it's not as advanced as ChemDraw, but it does the job quite well. And then there's absolutely no need for a third-party sketcher if you don't have a ChemDraw license or any other visual or like a 2D structure creator. All right, so then it's like this. It's very similar to the ligands that are currently have been crystallized with the crystal structures. So what we can do is we can give it a name. We're just gonna name it flexible docking tutorial ligand. So you see the name's gonna be changed here. And then you can go to file and then save structure as SDF. So we're gonna save it in the exact same way. Flexible docking tutorial ligand. And then you can close the sketcher, and then you can see that the SDF file has been created here, and I can open it with the name, and then you can see the structure has been created. Alright, all this is great. Now that we have it in 2D, what we're gonna have to do is we're gonna have to prepare it for 3D conversion, and we do that with convert. So we're gonna put here convert 2D to 3D flexible docking. We're gonna choose the 2D to 3D mode. The solvent should be water in case you want nitrogen's protonated and carboxylic acid deprotonated. Otherwise, choose organic solvents. We don't want to generate tautomers. We don't really have any tautomers in this anyways. And the molecule name is from the top of the file. Then we can click save on convert and then exit. And then what we want to do is we want to smart the file that we've just converted from 2D to 3D. So we choose here from convert box. We keep the name ligand smart. We don't want to run them docking on multiple cores because it's just one docking job, and then the mode is docking. So we save this, and you can notice that because we've chosen the ligand to be taken from the convert box, the pipe between prepare and smart has been removed. So then smart is not going to prepare the ligands for docking from the crystal structure. Okay, once this is done, we can do process. So again, it's already set up for you. It's taken from the prepare box above, all the aligned and mutated proteins, the number of ligands, everything is done here for you, and then the same with docking to flexible protein. So all of this is done. You just need to save the parameters. Alright, and the last step in the tutorial is to run it with fitted. So again, it takes all the information from the boxes of above. You should always double check though to make sure that everything's in order. Then the macromolecule is protein. The protein flexibility mode should be set to automatic. Once it has more than 2 protein structures, fitted will automatically determine that it has to do flexible docking. We're going to keep the crystallographic waters. We're not doing any covalent docking. The ligand file is the smarter file. We're not evaluating our MSD because we're not taking the ligand from inside the protein structure and then putting it back in. We're not running into multiple cores. We can put here flexible docking tutorial, and then the binding site and interaction sites are already chosen for you. Alright, so then we can click save, and everything has been created, all the keyword files. If you want, you can just check the keyword files for yourself in case you want to make sure that everything is written as you intended it to be written. Same for convert and all the other ones. They're pretty simple files, and they follow the exact same syntax that you've chosen in the GUI. And then what we can do is we can just run the workflow. So it's going to start with convert. So convert has already run. It's converted it to 3D. I'm going to show it to you in the meantime while the other processes are running. So you can see here it's added hydrogens, it's optimized the molecule. So now this molecule is suited to be run with smart. We are now adding hydrogens to the protein structures, and this has to be done to every single protein, so it might take a bit of time because we're doing 10 iterations of these randomized hydrogens, and then you have to do it 4 times because you have 4 protein structures. So but everything is running smoothly so far. It tells you at every single step where you are. So sometimes you might see different error messages for a specific protein, and that can be related to PDB naming that's not correct. Sometimes you have, for example, histidine amino acids that instead of being called HIS, they're called HSD or HSE, depending on which nitrogen is protonated, either the delta or the epsilon. So these names have to be changed. There's various other types of amino acids that can have different names from the common ones. For example, if you have a serine amino acid that has reacted covalently with a molecule, the name is usually not going to be serine. It's going to be some sort of hybrid of serine, like SGB. This also has to be changed back to serine. But Prepare is pretty good at telling you where the error or the problem is, so then you can go and manually fix the PDB. Unfortunately, this happens quite often with different PDBs because they're let's say that because they're sometimes tens of thousands of atoms, it's very hard to quality check all the amino acid names. The same for ligands. Sometimes the hybridizations are different than what they should be. Valences are different. They require deprotonation or protonation. So at every single step, I highly suggest you check the files that are being created just to make sure that everything's in order. For example, we would be looking at the ligand that is taken out by Prepare, and then you can visualize it and then go back. This is for 1E2K. So for example, what you can do is you can go to the 1E2K structure, and then you will see, if you go down the PDB, you can see what ligand should be there. So then you can compare it to the ligand that you have here. Now, this is just a visualization thing from Discovery Studio, but these are all double bonds. And this is also a double bond. You can pretty much infer that from the number of hydrogens and the flatness of the ring. But then you can look at it, you can check stereochemistry, and I highly recommend you do that for all your files. Always double check everything that's being created, and it will save you a lot of headaches in the long run. And it's also good to double check software in general, not only talking about Forecaster, but at the end of the day, it's code written by people, and people sometimes make mistakes. So it's always good to be on your toes and to always make sure that you have the correct things running. Alright, so all the binding side files have been created, all the protein files, and now we are currently docking. So here in output, as I've showed before, we create a log file that collects errors and warnings. We have an output file that tells you exactly all the settings, what's going on, and then what we have is an SDF file where we print out the doc poses, and also a results file summary. We also have an HTML file that gets updated once the first molecule is docked. You can click on all of these things. It auto refreshes every 15 seconds, so this might close as I talk. So you can see you're using 4 different proteins, and here are the names of the proteins you're using. This is the binding cavity you're using, and this is the interaction site and the first field file. This is the parameter file you're using, and then the ligand and however many molecules this ligand has. Usually whenever you do a beer screening and you have, let's say, 100 molecules or so, it's very easy to figure out how much time is left, because we automatically calculate this based on the average docking time each molecule takes, and we also update the progress as it happens. So I would run 2. If I open the summary, you'll see this is the best run with the best energy score and then math score. And then at the end, when all this is done, I'm going to explain to you what this field represents. And we're almost done with the docking. It's quite fast, even though it's a bit it's a very thorough algorithm. It's a genetic algorithm that does a very good job at sampling the conformational space. but this being said, it's quite efficient. You can also check the log file. For example, you can see here the matching score has been reduced by a specific number. This is based on the interaction sites that have been created by process. And usually you don't really need to care that much about, like, these aren't necessarily errors, but rather like things that are being collected during the docking process, which is now done. So now if you look here, docking has been completed, and this tells you, this only gives you the best run, and it tells you how long it's taken and what the overall fitted score is, which is a combination of both rank score and match score. I'm not sure if you can see my the pop-up that comes up once you hover over a cell, but it gives you all the information you need to know about every single of these cells. Okay, so once this is done, we can open the summary results file, and it will tell you a summary of the best run. So that was number 1. We take the best run as being the lowest in energy, so this one has an energy of minus 99.2 kcal, this one is minus 97, and this one is minus 98. Now what this specific field represents is basically which structure fitted chose to do docking. So if you see 0.000 for one of these proteins, it means that that specific protein structures has been chosen to do the docking. So in all 3 cases it was 1E2K. We can go again to the magnifier here, and then if you click on read numerical results, it will open this exact file that I told you about, so the results file. But then what we can do is we can open the poses, and again you can open them in the native JMol that we have here. So you have the interaction sites, you can see how well it fits over the interaction sites formed by the co-crystallized ligand, and then you can just open it in the protein and visualize the different interactions that this compound forms. You can also calculate hydrogen bonds, and it will show you what hydrogen bonds the compound makes with either water molecules or with other amino acids. I can also show it to you in Discovery Studio. So forecaster tutorials, we can go here, output. So we have this, and we just copy it into the structures that we created here. So what we're going to do is we're going to hide the ligand. There we go. That's the second one. We can hide the ligand, and this is our dock ligand that is still here. Alright, what we can do is then we can center this. We don't really need all the hydrogens, so what we're going to do is we're going to just show polar hydrogens, so that's great. Then what we can do is if you click Control E, you're going to get this little menu, and then you can select amino acids that are within 5 angstroms of the ligand, and then you can go to display style, and you get this little menu here, and you can choose to show them as sticks, and then all the amino acids show up here. So these are the amino acids that are within 5 angstroms of your ligand. Usually what I do is I change the color of this to either red or like a bit pink. Another thing that I do to help me visualize this is I first hide this, then select the entire conformational ensemble, then I go to structure surface, and then I add a solvent accessibility surface close type. So now I have the protein as a surface, and because I don't want all the ribbons and everything, because they just add noise whenever I try to visualize stuff, I can go to protein and then just stop displaying. So now what's gonna show me is all the water molecules and all the amino acids within 5 angstrom of this ligand. And then what I can also do is I can go to non-bonded interactions and then choose all the interactions that I want, usually intermolecular between molecules. And then it's going to show me all the interactions that my dock pose makes with the amino acids in the binding site. And if you click on the reason I keep on toggling on and off the solvent accessible surface area is because if you try to select anything, it will just select the surface area. But if you click on this, it's going to tell you what type of interaction. There's also a cool thing that I think I might find here if I'm not. Oh yeah, okay. So what's really cool is you can choose. Let me see if I can find it. Yeah, okay. So if you click on non-bond here, it's going to give you all the interactions that the ligand forms with your protein. What type? Whether it's hydrogen bonding, whether it's halogen bonding, pi stacking, pi sigma, pi sulfur. It's a very neat way of visualizing dock poses, and I use it very often in any sort of project. Another thing that you can do if you're feeling very fancy is you can also add a surface to your ligand to see how it actually fits. So instead of putting solvent accessibility, you can go to custom and give it the same color as your ligand. So then when you put in the solvent accessible surface area, you can see exactly the space that it occupies inside the binding site. Alright, so you can see the different, especially here on this glutamate, that they're different, for at least for one protein, that they're different conformations. So that's quite important because it doesn't form a hydrogen bond, or I guess this oxygen forms a hydrogen bond with the ligand. But yeah, so you can use this sort of conformational ensemble docking to see in which of these protein structures your ligand would fit the best, and it's also a very good tool to figure out the flexibility of your protein structures, because as I said at the beginning, sometimes you have multiple protein structures that are all good. They have good resolutions, good B factors, water molecules are resolved, and you don't want to lose that information. So this is a very good way of using that information and of doing flexible docking. I hope you enjoyed this tutorial. If you have any questions, you can reach out to us at, I'm just gonna put it here, at info@molecularforecaster.com. You can also leave comments. You can find us on LinkedIn, also on our YouTube channel, and we would be very happy to help you in your all your inquiries. And I hope to see you next time, when we'll most likely talk about cross-docking, which is, in my opinion, one of the most important types of study that you can do whenever you have multiple protein structures. Until next time, I wish you all good health and good fortune, and I can't wait to talk about cross-docking in the next tutorial. I wish you all a great day, and I'll talk to you soon.